Hey everybody, it's Mr. Z. Today we're going to be talking about the central dogma of biology, and I'm going to do a little chalk talk to give you a review and summary of what this is. So the central dogma of biology is a hugely important process that is at the center of every cell's functioning, and without these processes that I'm about to tell you about, life would not be possible. And at the heart of all of this is going to be DNA. So we know that DNA is the blueprint for life, and the essential dogma helps explain why that is. So DNA can do one of two things. It can either replicate itself with the help of some other enzymes, and when it replicates itself, it's just making a second copy that, for the most part, is 100% the same. Unless some mutations arise, they're identical copies. And this is so that when the cell divides, each of the two cells has a complete copy of our DNA. Because if the cell divided and we didn't have replication, we would have two cells that only have 50% of our DNA. And then they divide again, and we only have 25%. Honestly, after one division, our cell probably wouldn't even be able to function. So replication is key for the ability for cells to divide and grow and develop. And if you're a multicellular organism, that's probably something that you would like. So replication is very key for our survival. And the other thing it can do is it can get read and get copied into something called RNA, which is like a sister molecule to DNA. And this process is called transcription, and it is done by a special enzyme that is called RNA polymerase. And RNA polymerase is going to go through and it's going to read the base pairs of our DNA. And what happens is it will look at, the A, the C's, the G's of the DNA and then find corresponding RNA bases that match up and base pair with those to create this sister template of RNA. Now, why would we want to do that? Well, there's multiple reasons, and I'm going to get into that in a minute. But what I did want to bring your attention to is that this process doesn't occur on our entire strand of DNA necessarily. Remember that there could be thousands of genes on a single strand of DNA. And so what happens with transcription is actually that just one segment of it might be transcribed. And that segment is going to be called a gene because it codes for something special like a protein. So a gene is going to be transcribed, and it's only part of this DNA. The gene, the RNA polymerase will start at the beginning of the gene, and it's going to find matching base pairs of RNA, and it's going to polymerize those and create a nice little transcript. So then this transcript is going to go out into the cell, into the cytoplasm, where it's most likely going to interact with an enzyme called the ribosome, and it's going to be translated. Now, translation is what happens when we go from a nucleic acid to a protein, which is made from amino acids. And translation occurs when the ribosome will actually bind to the RNA, and it's going to start looking through the RNA and finding something called codons. So codons are actually sets of three base pairs. If we imagine these are each three different bases, it's going to look at these three, and then there's going to be a little molecule called tRNA that's sort of like the Rosetta Stone of molecular biology, I guess. And it's going to come in and it's going to match up with these three, and it's going to have a corresponding amino acid on it. And the ribosome will take that amino acid and it's going to polymerize it through a dehydration reaction and make a longer polypeptide chain. We're going to get into all of these little details later on, but I wanted to make this video so that you could always go back and reference it. So you may not know all of this yet, but just keep in mind we will be learning it. Okay, so we've got our ribosome making these polypeptide chains. Once they all get completed, they're going to get folded up and turned into a wonderful protein. And proteins can go out into our cells and do all sorts of great things. So, for example, I want to take a look at the enzyme called lactase, which is important for digesting milk and milk sugars. Lactase is a gene that would be found in our DNA. So let's see, let's make this pink. And so lactase is a gene that's found in our DNA. There's a lac gene, and that gene will go ahead and RNA polymerase will read through that gene, and it'll make an RNA transcript. The RNA transcript will then get translated by the ribosome, and it's going to end up making a little enzyme that chews up all of our milk sugars. So I'm going to make them look like Pac-Man because they basically go around and they break lactose sugars up. Okay? Now, a couple things to point out about each of these processes along the way. One is that the reason that we like to have this intermediary or RNA is that it offers us a couple very important advantages over just copying and turning in DNA into protein. And one of those that I haven't mentioned to you before is that the RNA helps us amplify genes. So you could imagine that in our genome, we only have one copy of this lactase gene. So if I needed to make a lot of lactase in my cell because I'm drinking so much milk, there might not be enough time to get enough lactase enzyme out in time to digest all of that lactose sugar, I should say. So what RNA affords us is the ability to amplify. So I could go through and transcribe 10 RNA transcripts of lactase, and then those 10 could spit out a whole bunch of lactase enzymes. So instead of having, let's say, instead of having 3, I could make a bunch more lactase enzymes because the RNA transcript allows us to amplify the gene. Okay, so that's one part of it. The other part is that RNA helps us protect our DNA. And that's because we don't have as much machinery moving in and attaching to our DNA and interacting with our DNA. The less stuff we have coming in to interact with our master copy of all of our genes, the better it is for us. Because we don't want that to get damaged. We don't want our DNA damaged. We don't want it to be mutated. And so this way we can make a few copies of an RNA transcript, and then that RNA can get processed many, many times to make lots of enzymes. So it reduces the amount of impact of damage to the DNA. So the RNA leaves the DNA, goes out into the cytoplasm, it gets processed by the ribosome, and at this point, now we have all of this lovely lactase enzyme floating around ready to do its job. So let's imagine that I drink a bunch of milk and we get this milk sugar coming in called lactose, and luckily for me, I am lactase persistent, meaning that my lactase gene is turned on and I've got all of this lactase enzyme in my stomach. So they're going to go ahead and they're going to chew up all of this lactose sugar, and Mr. Z is going to be a happy person. I love my milk. Now, in some populations and in some people, they are not lactase persistent, meaning that they do not have lactase into adulthood. And we call them lactose intolerant. You probably have heard that before. And what this means is generally that they actually have a copy of the gene in their bodies, in their stomach cells and whatnot. However, it gets turned off. So this lactase gene is going to get turned off. Let's see, let's make that blue. Okay, so it gets turned off and it's no longer getting processed, which means that our RNA is no longer going to get processed, which means that we're no longer going to have a protein getting processed, which means that we no longer have our lactase enzymes coming in to break up that lactase sugar, which means that all this lactose is going to go into our stomach. It's not going to get broken down, and it's going to cause us a lot of gastrointestinal stress, which means that we are not happy campers. We are actually very sick feeling and not too happy at all. So this was a quick rundown and an example of how the central dogma might have played a factor in your life already. If you can drink milk, that's because you've got this lactase gene turned on, and it's getting turned into RNA, and that RNA is getting translated into proteins. If you are lactose intolerant, that probably means that your lactase gene is turned off, and the transcription is not occurring. That's what turned off means, is that the transcription is not occurring. And without any RNA, we can't possibly have translation because we don't have the messenger template. And with no translation, we have no protein, and we have an unhappy person there when they drink or digest any dairy products. So there you have it, central dogma of biology. It's important for life, and it's important for drinking milk. All right, have a good day. Thanks for listening.